This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 322, recorded on January 30th, 2015. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today here in the TWiV studio, Postcards, Dickson, Despommier.
Barely. Hello, Vincent.
You know what postprandial means?
But of course.
Did you have a good prand?
Exactly. First I pranded, and then I posted.
Nice day out, isn't it?
It's gorgeous out there, actually. For what it was a couple of days ago, it's really nice.
It's cold, though.
It's moderate right now, but it's supposed to get really cold tonight.
2 degrees Celsius. It's going to minus 15 Celsius tonight.
Exactly.
But it is sunny with just clouds floating around, right?
That's right.
Also joining us today from southeastern Michigan, is that right, Kathy Spindler?
That's right. Hi, everybody.
Why do I cannot remember it?
I don't know.
Getting old. How are you?
I'm fine. I used to live in the southeast U.S. Now I just live in the southeast of Michigan.
So all people from Michigan, when they tell you where they're from, they just use their hand.
Of course.
And then they point to a region of their hand, and you're supposed to know where that is.
Yeah.
All right. I'm looking at Michigan here on the map.
I know where that is.
Yeah, Ann Arbor is near Detroit. Yeah, it's southeast. Maybe now that I looked at it, I can remember it. Southeast. I look at right in the middle there, Lansing. Mm-hmm. After which Lansing polio virus was named, that we worked on for many years.
It's about 30 miles north of South Bend.
Yeah, where you were, right, Dickson?
That's correct.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
In the West. How's the West?
Yes, the West, although I'm east of... actually, I guess I'm east of all of you.
You are.
Yeah.
Yeah, it's not too bad. It's 34 Fahrenheit, 1 Celsius. It's supposed to get down to really, really cold tonight and kind of stay that way.
How did you like the blizzard of historic proportions?
It was annoying, but I wouldn't really call it historic.
That's what the weathermen were saying.
I would say
we
hysterical.
got about... I guess we got a foot, a little over a foot, maybe a foot and a half.
You guys got more than we did.
You got a lot.
It was very, very powdery, kind of annoying to shovel because you flip the load of it up with the wobble, of course, and flip the load of it up onto the pile, and it would kind of slide back down again.
So Mount Snowplow doesn't exist this year?
It definitely does. There's Mount Snowplow is for real, but the summit of it is getting a little bit tall.
Kathy, was the blizzard predicted to hit you guys?
Oh, not at all. No, we were fine.
Yeah, they were predicting like 30 inches of snow here.
Remember, Kathy's a flatlander.
Yeah.
Well, my sister down on the south coast of Massachusetts got about 2 feet, I think.
Exactly.
So somebody got 2 feet.
Oh, yeah.
Yeah,
And
Boston.
So did Montauk Point.
Is that right? Yeah.
Yeah,
and Nantucket got slammed.
I'm sure.
So there was a storm somewhere.
There was a storm.
Thank
There was
goodness.
Nantucket lost electricity. They had feet of snow there, and they don't normally get the blizzards that we get, so a little unusual for them.
Yep.
Well, there is snow on the ground here, a couple inches. It's kind of nice.
We have another sunny day.
Yeah.
It's 20 Fahrenheit and minus 7 Celsius, so it's really nice.
Is
really
it like
pretty.
every TWiV Friday is a sunny day, huh?
It's working that way, yeah. And in case any cartographers are going to get on Dickson's case, Lansing looks like it's about 60 miles north of South Bend and 45 or 50 miles east. So it's a 2-hour, 20-minute drive, 156 miles total.
All right.
So
Today, for your listening pleasure, we have all email. No, don't shut it off.
We've got some good ones.
Don't touch
I
that
know some of
dial.
you don't like all email episodes, but we only do like 1 or 2 a year, right? Less?
We're way
we
behind.
do a few more than that, but we are behind.
And there are some good things here. People suggest articles, and we talk about them, so stick with us. We have some follow-up from last episode. Laura writes, "Hi, TWiV team. I was alarmed to see that Mark Lipsitch published his views on potentially pandemic pathogens in Scientific American. It's too bad that the readers are getting only one side of this argument. If you are already aware of this, please disregard my email. This came in just a day or two ago, so Laura has not listened to TWiV 321 where we talked about that article. I can't thank you enough for your tremendous science outreach. The air in Salt Lake City currently contains 33 micrograms per cubic meter, particulate less than 2.5 microns. With air quality this bad, who cares about temperature, dew point, etc.? With great admiration, Laura.
Wow.
I like that because no one ever...
We should probably frame that one.
Yeah, they don't give you the particulate count. Yeah.
They have problems.
The comments in the Scientific American underneath the article are a bit annoyed with Scientific American for publishing the article. So...
Mm-hmm. Well, I think you should publish both sides. If this is obviously someone's opinion, right?
Right.
And it almost looks like this is an official position of SciAm.
Right.
Joe Ellen writes, Dearest TWivers, thank you for being part of my life the past 4 years. I realized I was behind listening this winter as I live in Minneapolis and am usually either weeding or shoveling enough to stay caught up with all three and NPR podcasts as well. I read the Scientific American article referenced in 321 and realized just how behind I was when I went immediately to TWiV for the rebuttal and had 3 more in between to listen to. It's currently 16F and Starry. I like that we are...
I like Starry.
Starry is cool. We're put together with NPR podcasts. Right?
Yeah, that's nice.
I have been reading William James' Varieties of Religious Experience, and directly after listening to TWiV 321 came across the following quote and decided I must finally contact you. So here it is. Quote, if any of you are enemies of what our ancestors used to brand as enthusiasm and are nevertheless still listening to me now, you have probably felt my selection to have been sometimes almost perverse and have wished I might have stuck to soberer examples. I reply that I took these extreme examples as yielding the profounder information. To learn the secrets of any science, we go to expert specialists, even though they may be eccentric persons and not too commonplace pupils. We combine what they tell us with the rest of our wisdom and form our final judgment independently. End of quote. Thank you for being some of my favorite eccentric persons. While I would love to meet many of you personally, I will enjoy your company via Twix. Cool. I like that.
Yeah. I like that quote.
That's very good. So are we eccentrics? What is an eccentric? Let's have a definition.
It's probably 2.5 standard deviations from the mean of whatever normal behavior means.
That's funny that you would think of the standard curve because that's exactly what I was thinking.
Right.
Which side of that standard curve?
I was thinking of something that was not rotating around its center. That was rotating.
So would you like the noun or the adjective?
We're ellipsical then.
The noun, a person of unconventional and slightly strange views or behavior.
Strange.
Here's mine, a disc wheel mounted eccentrically on a revolving shaft.
There we go.
Yeah, well,
Unconventional and slightly strange behavior. Well, I guess that Joe Allen thinks we're eccentric, so we must be.
I've been accused of that before.
Yeah.
All right, and the final follow-up is from Sandra, who writes, latest TWiV. Paul Dupree is an incredible diplomat. My indignation button is much more sensitive than his, and his cautions in the latest TWiV about how scientists speak of A-trip. He actually uses the other word. But Sandra, come on, A-trip. Right. And about how people hear those scientific remarks seemed very reasonable. I honestly felt a bit guilty about the intensity of my negative opinions about some of the scientists and ethicists who have commented on A-trip. I don't feel guilty, Sandra. R.E. Relman. So Sandra had originally commented last week that she was disturbed that Relman had said safety has to be now, or rewards have to be now and not in the future. About
benefits, yeah.
Benefits.
Right.
So she's revisited that. R.E. Relman at the December A-Trip Symposium. Session 3 began with Yoshihiro Kawaoka's presentation, which wowed me when I first saw it in December, and wowed me again tonight watching the video of it on YouTube. Tonight I don't have pneumonia, so hopefully my brain absorbed more of it this time. Among other things, Kawaoka put up a slide showing Lipsitch, and Galvani's 3 recommended alternatives to ATRIP work, and then a number of slides explaining why each alternative is inadequate. He concluded, quote, "We cannot rely solely on alternative approaches," end quote. Incredible presentation, so much factual, relevant data presented so succinctly. Relman comments start at 17:35 on the video. At 28 minutes, Relman speaks about how benefits of ATRIP must be immediate. And she actually sent a capture of his slide, which he put up at the presentation, where it says that basically. And we'll put that on the show notes. At 47.25 minutes, Harvey Feinberg very politely called out Relman's stated concern about ATRIP resulting in the development of a virus that is simultaneously highly pathogenic, transmissible, and resistant as, quote, kind of an extreme straw man, unquote. Excellent, Harvey Feinberg. As a non-scientist, I would never engage in an argument with anyone about the ATRIP dispute, but as a science-curious and analytical human, I feel much more confident in what I hear from you and from Kawaoka than from what I hear from Relman and Lipsitch. Sandra. P.S. High temperature today hit low 70s F and forecast highs for upper 70 later this week. This would have been a great week for you to interview a scientist in Texas. Yeah, not by Skype, though. Yeah, I saw what she had written last time was correct. And, you know, Paul was saying, oh, maybe it wasn't so and so forth, but it was so. But this is...
So to interview a scientist in Texas, we should have taken a trip.
You bet.
Ooh.
This is the thing, you know, all this extreme straw mans or straw persons set up. It's crazy to make your point. All right, so now we will read some emails. And let us start with Alan.
Okay, John writes, in TWiV 319, Alan picked a book on making a toaster from ingredients. The toaster tale feels like an update of the classic essay, I Pencil. No one person can make a pencil. The process requires a complete economy with hundreds or thousands of kinds of specialized labor. And he gives links to the original I Pencil essay from 1958 and a Wikipedia article about the essay and its impact. For the younger listeners, pencils are an archaic weapon once carried by students to ward off attacks by standardized tests. They came in only one kind, inexplicably called number 2. Well put, John.
I actually had to buy number, 6 pencil once for something,
Really?
and number 1 pencil. So
I took a drafting course in high school, and that was when I learned that there was a whole collection of pencils. And
Oh, yeah.
yeah.
Well, I took a course at the Art Students League for a while and carried a complete set of pencils with me. Number 6 is very soft.
Yeah.
Number hard will etch a hole through your paper.
Number what?
Number 1.
Yeah.
I mean, it's really, really hard.
I love mechanical pencils.
Yeah.
I absolutely love them. Regular pencils they wear down, right?
Yeah. That's right.
Dickson, can you take the following from Igor?
Do my best.
No, more than your best.
I'll do my best. That's all I can promise. So Igor writes,
Such an underachiever.
Dear Vincent and friends, I want to praise you for doing this immunology-heavy paper as part of a virology podcast. I'm a virologist by training myself, but at some point my work shifted to HIV vaccines. So I had to quickly learn some immunology, and it gave me a whole new perspective on viruses. Studying viruses in cell culture is great fun, and one can certainly learn a lot, but one always have to remember that these are artificial systems that minimally reflect the complexity of the environment in which the virus has to survive and propagate. Antiviral host responses are the major driving force behind viral evolution. If I might play on the famous quote, "Nothing in virology makes sense except in the light of immunology," please do more papers like that. Best regards, Igor.
Who said that? Does anyone know?
I don't know that.
I would say nothing in immunology makes sense except in the light of virology or microbiology.
This is a play on another quote that I think it's nothing. I think it's something like "Nothing in biology makes sense except in light of evolution."
I tried to Google it, but I got too much. Yeah, let's try it again. Nothing. Google, copy, tab, paste, return. Nothing in genetics makes sense in light of nothing in biology.
Nothing in biology makes sense except in the light of evolution. It's an essay. by
Jerry Bergman.
Huh.
Does that make sense? Anyway, thank you.
This is the Wikipedia at least cites Theodosius Dobzhansky.
Oh.
Okay.
But the quote, I have seen the quote floating around. I don't know who the originator was.
Yeah, we'll try and do more immunology. I would like to do that, Igor.
Yeah, is he referring to the TWiV 316, I think?
Yeah, that was the one about, that was about an alpha virus neuro.
And the IL-17.
Yeah. Did you miss that one? Oh, you were here, right? Yeah.
Yeah.
You were here. 316, exactly. Yeah. Kathy, can you take the next one?
Sure. Johan writes, 3 degrees Celsius, humidity 86%, wind 3 kilometers per hour, and light rain here in Sollentuna, just north of Stockholm, Sweden, at 23:30. BBC NewsHour is BBC World Service's premier news show for the international audience, and today they are doing a single-topic show on Ebola. This is the first time I've heard them do a single-topic show, so I think they are quite rare. I certainly haven't heard one before, and I listen daily since a few years back. It gives the link to the NewsHour Extra about the Ebola response. Owen Bennett-Jones is joined by a panel of this BBC's expert correspondents in West Africa and beyond for a special program assessing the international response to Ebola in West Africa. And then he goes on and it lists the other correspondents and so forth. And so it's something that you can go back and listen to. And I'm pretty sure I caught at least part of this because I'm often awake at odd hours and listening to the BBC World Service. So I know Owen Bennett-Jones and, lots of these other BBC announcers.
Right.
So it was good what I heard of it, and we can all check it out.
You wouldn't call that eccentric behavior, though, would you, Kathy?
No, I call it insomnia.
Right.
And we have a show title.
Right. That's right. Is anybody out there sleeping?
He writes at the end. Oh, you can read that.
Thank you so much for TWiV, TWIM, TWIP, etc. Blah, blah, blah.
Blah, blah, blah. I listen to a podcast called Security Now, and they often read questions from listeners, and they bypass all the praise. We read our praise, and they always go, "Love the show," blah 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 blah. And I'm always like, "Why would you say blah blah?"
Et cetera.
All right, so we're around to me. Don writes, "Do you know the number of infections compared to the clinical symptomatic infection rate? Thank you for all the wonderful TWiV series." And I forgot to. Yeah, it was Ebola.
This is Ebola, yes.
It was in the subject, but I forgot to. Well, so I went over to, what is it called, the WHO page, stat something? SITREP.
Oh, yeah, that's right.
So there are 22,101 cases of Ebola and 8,818 deaths. That's 40% case fatality ratio. But if you use laboratory confirmed, so not all those are laboratory confirmed, only 13,000 and 800 over that, so that raises it to 64%. So the 40% number has gone down since the beginning, remember? And I think that's partly because of the better care that the patients are getting there, right?
Sure.
Right, and so the question, I guess, was symptomatic versus fatal. Because as written, I initially read this as the number of infections compared to the symptomatic infection rate.
Number of infections. Well, we don't really know that, right?
We don't really know asymptomatic infections. We've cited a few studies over past TWiVs that talked about seroprevalence, and there may be asymptomatic. There probably are asymptomatic infections out there, but we don't know how frequent they are.
Yeah, there are. Yeah, we don't know because you'd have to do a serological survey afterwards, and not clear that we're going to be able to do that.
And even that doesn't give you a perfect answer. But yeah.
All right, we're back to you, Alan, with Jeffrey.
Okay, Jeffrey writes, "Doctors, I thought I might pass along a small but potentially interesting article I ran across. And this is Pumala hantavirus infection alters the odor attractiveness of its reservoir host. Published in Ecologia in 2014, provides the DOI. I suspect it's behind a paywall, but I'm not sure. Anyway, the gist of it is there seems to be a small but detectable and temporary aversion among healthy bank voles to the urine of bank voles infected with Pumala hantavirus. It is erratic but measurable. It has interesting implications in both transmission modeling and viral manipulation of community, though not necessarily host behavior. Beyond that, however, is the interesting idea that we might be able to run some relatively quick diagnostics by volatile analysis of urine. I know there are medical conditions where the color and odor of urine is diagnostic, but this points to the potential for using urine volatiles analysis as a more powerful diagnostics tool, unless this is already used for the detection of specific diseases and I didn't know about it. There was also mention of an interesting reservoir for polio and norovirus that you might not have noticed. In another article, this is from Journal of Applied Microbiology, localization of norovirus and poliovirus in Pacific oysters. Any thoughts on if and how this might affect polio eradication campaigns? I assume that the virus flushes through oyster beds within a generation or so, but I don't know. Also, given how often polio was contracted at the beach, has this reservoir been noted before? Thanks for the informative podcast, Jeffrey.
Dickson, do you remember the couple of parasite TWiV papers we did about odor alteration by parasites?
Yeah, we did one on malaria.
There's one, the most recent one was odor rodent.
Odor rodent, that's right.
Vincent and Dickson discuss how malaria parasites induce odors in their rodent hosts that attract mosquitoes.
Exactly.
And then the other one was you're in a game. This is a good title. You're in a game of cat and mouse.
Right.
URINE.
Yes.
Infection with toxo causes mice to lose their aversion to cat urine.
That's right.
Right.
And also this reminds me. I know this wasn't Jeffrey's point actually, but yeah, you guys may remember we did a paper a long time ago where there's this insect virus transmitted to plants. And I think it's an aphid-borne virus or something. Anyway, the plant gets it, and it alters the odors put off by the plant.
Yes.
Right.
Then that attracts more aphids, and they bite, and they hate the taste, and they immediately run off and bite another plant.
And that helps spread the virus.
Isn't that cool?
Yes.
Yeah.
But it's about odors. So that's a good point. Maybe urine would be a good place to look for volatiles that might be diagnostic. Do you know anything about that, Dickson?
I don't.
Okay.
But I do remember an article where the tobacco hornworm was chewing away on a tobacco plant, and the odor emitted by the injured tobacco plant equals the pheromonal signal for the parasitic wasp to come in and lay their eggs inside the hornworm. So
yeah.
Odors do play a huge role in nature in terms of maintaining balances.
Yeah, I don't think this has been looked at very much. The idea of using urine that way as a diagnostic. Go ahead.
Yeah.
Well, I was just going to say that this story with the bankfuls, though, seems to not favor viral transmission.
No, not at all.
Seems to be
the other way,
favoring antiviral transmission. So
right,
that made me think that it, is something related to innate immune response or something like that.
And I wasn't able to access the paper, but it would be interesting to know if they've looked at other diseases in these voles. Is this a general aversion to the urine of diseased voles, which would be a very adaptive response? If somebody's got an immune response going on, maybe that makes them smell bad.
Yeah, the transmission issue is interesting. It doesn't make sense, but then we're looking at it from a human perspective, and who knows what's going on here?
Our noses are not very good.
Yeah,
at
we
that
apply
stuff.
this. Well, if it smells bad, they're not going to transmit the virus. But who knows? Who knows? Now, this polio paper is just, they took oysters in aquaria in the lab, and they put polio and norovirus in the water, and it got concentrated in oyster tissues, okay? So the implication is that in the oceans, that if there's polio going in via the sewage, it might get, concentrated in the oysters. If you're immunized against polio, it doesn't really matter. Most of the polio around that would be there would be vaccine-derived polio. Of course, it would be virulent, but I'm not sure it's much of an issue for eradication. I don't think it lasts very long.
And if you have sewage-contaminated oysters, you're going to be more worried about other stuff.
I'll say.
Oh, yeah. Yeah, it's not going to just be polio in there.
No.
In fact, we know hepatitis A virus gets into oysters.
Well, you got cholera and stuff like
that,
too.
Never. So the point is, you just take your oyster and drop it into a shot of vodka before you eat it. Right?
In the South, we all deep fry them.
Dickson, I was at a ASM meeting in New Orleans once, and they were serving that at the bar.
Yeah, raw?
Yeah, raw oyster in a shot of vodka.
Yeah, oyster shots.
Is that what they're called?
Oyster shots. And sometimes there'll be a layer of Tabasco sauce on the bottom.
Oh, yeah.
So you get your vodka, your oyster, and your Tabasco all at once.
Now, before you start writing in, I understand that will not inactivate polio, especially in the deep tissues inside. Okay,
This is all true.
just joking.
Yes.
Dickson, you're next.
Okay.
Do enough of them and it'll inactivate you.
That's right. Christopher writes, "Greeting TWiV.
That's a new one.
That's a new one. That's
I
a
like
TWiV. So I have a question about a possible approach for an antiviral. Is there a way to produce a particle that at one end binds to a glycoprotein, parens, or anything on the surface of the virus, really, close paren, and on the other end simply has a piece of an antigen that we currently have a successful vaccine against? Is this a viable option to make the virus visible to the immune system? Has this been tried? It may provide a way around a couple of existing challenges of passive immunity. For example, ZMapp has had to find a way to humanize the mouse antibodies. Instead of having to do that, you could utilize the existing antibodies. The downside might be that antibodies get to the particle before it and binds to the viral particle. Any thoughts? I'm including a crude hand-drawn diagram just for kicks." Okay, TWiV.
I don't see why that would neutralize the virus, though. If the antibody is just going to attach to this known antigen,
Right.
That's not. You really need to bind to the particle to neutralize its infectivity.
Mm-hmm.
I mean, well, first of all, I appreciate the drawing, which says at the bottom, not to scale. That's wonderful.
It's good.
But I guess if you got a tight enough binding between this adapter protein and the viral particle,
That's the key. Yeah.
then you'd get the existing antibody. But antibodies are already kind of doing this, right?
So now you have an antibody kind of indirectly bound to the particle. It's not going to neutralize it because it's not binding to the particle. So what's going to happen next?
It'll opsonize and maybe get taken in by a cell.
Yeah.
I did think that for his binding molecule, he could use the FC portion of an antibody. Or wait, I don't mean FC. I mean the FAB, the antigen recognition part.
Right.
Yeah,
The FAB. Yeah.
FAB, yeah.
But if you can generate an FAB against the antigen, then why wouldn't you just develop a vaccine that does that?
Right.
Yeah.
Right.
I do like your icosahedral virus, though.
Yes. It's a nifty idea, and I like the drawings.
Yeah.
Those are our thoughts. We're not. If you were our student and came to propose this experiment, well, you could try it. If it were simple enough, I'd say, try it.
Yeah.
Why not?
You must have one of those pencils that's blue on one end and red on the other.
Yeah.
Is that a number 2 or number 6?
I don't know.
Kathy, you're next.
Okay.
Jody.
Jody writes, "Hi, TWiV team. Just for your interest, a paper has been published on the link between one of the H1N1 vaccinations used and an outbreak of narcolepsy among Finnish children during the swine flu pandemic." And Jody gives links to a PLOS ONE paper and I think something that's more like a press release. The findings are related to differences in viral proteins between the different vaccination preparations, Pandemrix used in Europe and Arapanrix used elsewhere. It was previously thought that the narcolepsy cases may have been caused by one or more of the adjuvants. In Finland right now, it's dry, partly cloudy, and -1 degrees Celsius.
I bet you it's dark.
Yeah.
So this was an increased rate of narcolepsy in younger people who had been immunized by the H1N1. This is way back when the 2009 H1N1 pandemic started. Right. I remember we talked about this. We said, we're not sure about the data, but it turned out they were good. It's real.
Yeah, there was a real. It wasn't a huge number of people, but it was statistically significant.
It also happened in France, Sweden, and Finland, right?
Right.
And in Norway, Ireland, and France and the UK. So more than just Finland. But yeah, it's interesting. There was originally proposed it might be the adjuvant, but it's not. They showed that in this study. It's something to do with differences in the protein composition, which, so they say, we are still working on this problem. We don't know how this would happen. But
why just in Finland, though?
No, it's not just in Finland. It's in other countries.
So
as
it
I
overlaps exactly with the distribution of the two different vaccines?
That's my impression.
Yeah. Pandemrix versus...
Goodness. Okay, why just Scandinavia?
Right. Well,
France.
Pandemrix was the only one that caused this, and it was only seen in Northern Europe. But I don't know if Pandemrix was used in Southern Europe.
Yeah, so they're comparing the Arapanrix with Pandemrix, and they originally thought it was the difference in adjuvants. But it's not. They ruled it out by this study. But it's not just Finland, Dickson, as I said before, and which you conveniently ignored.
Right.
Just kidding.
Is the vaccine portion related to the H1N1 the same in both vaccines?
That's the point. There's higher amounts of structurally altered viral NP in Pandemrix than in Arapanrix.
I see.
And there are detergent-induced antigenic changes of the viral NP that are recognized by antibodies from children with narcolepsy, and an increased antibody response to NP in association with a risk allele, a genetic component for narcolepsy.
They're made by two different companies, right?
Right.
So that's not surprising that you would have differences. Yeah. Well, we will await the solution of this issue, and then we will talk about it. I think it's an interesting finding. I'm not sure they'll find it out very easily. It's tough to do, and we're getting farther and farther away from the actual time. But it goes to show that you cannot predict what's going to happen when you put vaccines in lots of people. Right?
This is true.
Yep.
Anthony writes, he sends us articles about an avian influenza virus responsible for outbreaks at poultry farms in southwestern British Columbia. I have to look at a map to see where the heck that is. But anyway, it's H5N2.
It's probably near Vancouver.
Yeah.
British Columbia. Let's see.
North of Washington State.
That's right.
BC. Yeah, there it is. My son was just there skiing over his break in
Nice.
Whistler.
in Whistler. Yeah.
Ah, yeah.
He said it was amazing, Dickson.
Great place.
Yeah. Southwestern British Columbia.
Trout fishing
Got it.
ain't so bad either.
No.
No, it's
Anyway,
pretty
this is
good.
an H5N2 strain, which is highly pathogenic for chickens. That means it kills them. So it's a concern for the poultry industry, not a concern for people. That doesn't really. I looked into this a bit, and there's some controversy of whether this strain actually infects people or not. So we're not sure. But if you're a chicken or a chicken farmer, this is a problem.
Beware. That's right.
If you're a chicken, be afraid.
But great title. Just goes to show these influenza viruses.
If you're a
are
chicken,
problem.
live up to your name.
We have chicken farms with thousands and thousands and thousands of chickens all in one place, and it's a great place for these infections to get transmitted to from wild birds, right?
Yeah,
Yep.
you bet.
I don't know what you can do to prevent it. You can grow chickens and chicken tissue and culture, I guess, and that would get around it. If it tasted good, why not, Dickson? Right?
It's probably going to be 20 times more expensive to do it that way.
Yes.
Yes, you're right. Back to Alan.
Varun writes, "Greetings, profs." PROFZ. I like that.
That's another possible title.
More recently, I have noticed credibility of peer-reviewed publications has come under attack from scams and lack of trust. I wish to know what the comments are by the Twix team. Please find links below. And Varun links to. Bring these up again. I think.
Oh yeah, this is the whole Chinese paper mill scam.
Oh
thing.
yes. So this was
One guy reviewed his own paper.
Yes.
They got suspicious
so this
because the reviews came in within a day, and they said that never happens. I thought that was funny.
Yeah. So and there's the Nature article goes into this in pretty good depth too.
There's a Siam and a Nature article.
Yeah.
Well, basically, we sort of touched on this before, that it's getting very competitive, much more than ever, and people are cheating.
Yeah.
When opportunities to cheat arise, cheating will happen, right, Dickson?
You bet.
So the interesting thing that I found out of this is that by searching for one particular phrase, they were able to find a bunch of these, and that's beggar's funnel plot. And beggars is kind of a chimera of two statisticians' last name, and there is no such thing as a beggar's funnel plot. So there's all these papers that refer to it, and so right off the bat, that tells you there's something fishy.
Yeah. So, Alan, are you familiar with these practices?
Well, I certainly was not familiar with these practices before this news broke. This is not something I'd ever encountered. But the basic concept of the scam, so the one covered in the Nature paper, was kind of broke with a particular researcher who got caught doing it. He submitted an article to a journal, and he suggested a few reviewers, and the journal went ahead and used those reviewers. Well, it turned out that those reviewers were actually aliases that he'd set up, and it all came back to himself, and he wrote the reviews and submitted them under the names of those bogus reviewers.
Well, that's the journal's fault, because they could have seen if
Yes.
those persons had any papers, and they would have learned right away that they didn't exist.
Which, ideally that would happen.
So as an editor, I often have suggested reviewers who I don't know, and I go into PubMed and see what they've published. Right?
Yeah.
That's all they had to do.
But at a lot of the journals, especially the high volume ones, they're not that careful. And, a lot of papers have been retracted over issues of rigging the peer review system. So,
yeah, you got to be careful. I'm sorry. No excuse. High volume, low volume, no volume. Just do your work. That's what you're there for, right? I mean, that's not going to solve the problem.
No.
The problem is the competitiveness and all that. But these are simple things that can be done.
Yeah, the stakes are so high at this point in this game that people are now resorting to practices like this. And if hacking the system will get your paper in a prestigious enough journal to get you tenure, that's a lot on the line.
You're going to get caught.
Yes, ideally people will all get caught, but certainly there's a risk of that. It's like people who fabricate their data. There are always going to be people who decide, make the wrong decision, and they're going to do something wrong, and a lot of them will get caught. But that doesn't stop other people from thinking, "Oh, well, I'm smarter than that. I won't get caught."
Well, that's right.
Well, Farun, those are our thoughts. We're horrified. It can be fixed to a certain extent, but the real problem is competition. But in any field, when there's competition, people cheat.
Yeah. I think the onus for this falls. These that came out most recently, this scams that were going on. I think most of these are a case of just the journals not keeping on top of things the way they should.
Yep.
So do you think ultimately Tom Brady will suffer from a deflated ego?
Dickson, what are you talking about?
Yeah, you don't follow sports, do you?
That's a football thing,
You
right?
don't follow sports, do you, Vincent?
Yeah, but what does that have to do with this
Cheating.
paper?
Cheating. Competition.
Is he cheat?
Well, he claimed that somebody on his side deflated the footballs to his specifications.
Okay.
So, yes.
All right.
Yes.
Dickson, can you
Yes.
take Eric's?
I'll be happy to. Eric writes, "Hello, TWiV crew. Lately, I've been listening to the earlier TWiV episodes while I eagerly wait for the new ones each week, and I just finished episode 57, Virology in High School. I was curious if there are plans on the agenda to do another episode like this one. At the time, the pandemic H1N1 strain of influenza was the major theme of the kids' virology questions, and presumably Ebola would be what's floating around in their heads now. But even so, I think it would be so rewarding, as it was in episode 57, to understand what high school students are thinking about viruses in the world today. The episode was actually very helpful to me. I was practically taking notes on how you answered questions in a way that a high school student could understand it. Being a scientist, in quote, in the family, the virologist no less, I was inundated with Ebola questions this past Thanksgiving by my aunts, uncles, and grandparents. Though many of them do have a high school education, the field of virology is very foreign to them, and of course, media outlets capitalized on that misunderstanding, intentionally or not, and many of them were actually kind of on edge about Ebola virus. While I may not have done a perfect job of explaining the current situation in West Africa, I do think I answered a lot of their questions and might have allayed some of their concerns. I think your episode 57 was helpful in the understanding how to approach discussing something a bit complicated to someone who doesn't necessarily understand biology/virology/epidemiology. I think it would be well worthwhile for another episode, like episode 57, to come out at some point in 2015. Kindest regards, Eric.
Well, that episode was really initiated by my son, who was a freshman in high school at the time. And my other two children have shown no inclination to do similar things, probably because they're... No, I will not say anything. So I'm happy to go to any high school who wants to have a session, but I don't have any particular plans. It was a lot of fun. They asked good questions and so forth, and I learned things. I would do it slightly differently next time.
Well, basically, Vincent, aren't you doing that for the undergraduates in the downtown campus? Because none of them have backgrounds in virology, so you have to start from scratch.
Yeah, but they have science. They're a little more sophisticated.
But you're not talking down to them, but you're reducing the terms to an understandable volume that they can absorb it.
But I think this was even more reduced.
Uh-huh. Below the level of actual usefulness?
No, I think it was useful. Why don't you go listen to it, Dickson? I've listened to every episode, you haven't.
You're so abusive sometimes. How come I don't take offense at these things?
You have a good nature.
No, because I know they're true.
Kathy, can you take the next one, please?
Eric writes, "Hello, TWiV crew."
Sorry,
that was Eric. Sorry,
it's okay.
I didn't move on down.
No worries.
John writes, he first of all sends a link about the Kansas man's death, and another link, Bourbon Virus is deadly, unlike anything seen in the United States. So this is a virus that killed a farmer in Kansas, and I don't even know how you pronounce this. Fagoto virus?
Yeah, that's it.
It's a Fagoto virus, which I just think is a cool name for a virus. So it's in the same family as influenza viruses,
Really?
The orthomyxoviruses, but evidently it's vector-borne,
Yep.
right?
Yep.
So that makes it a little bit different.
Like ticks.
Mm-hmm.
Oh.
The headlines are a bit much here. Unlike anything seen in the U.S.
Yeah.
Flu kills seriously, I don't see why. There's one guy who died, right?
Yeah.
And if it's a tick-borne disease, something like that, we've certainly seen plenty of those.
Yeah.
You bet.
In fact, we've seen the Powassan virus, the one we talked about, Heartland virus.
Yeah, Heartland. Yeah. Yeah. Well,
So,
if you just Google Bourbon virus, you see all the unlike any other virus, unlike anything seen. All the news sources have the same headline: deadly and unlike anything.
So is this a zoonosis that we're
Just
talking
to
about?
scare people. We don't know, Dickson.
Oh, I see.
But Fagoto has mammalian hosts, so not people. So it could be.
By the way, if you see the same headline or very similar headline reported multiple places, it means one of two things usually. Sometimes both. Either the wire service has picked that up and used it as their headline, or that was the headline of the press release.
Yeah.
Sure.
Yeah, they didn't do any changing, right?
Yeah.
So ticks have specific host preferences.
Yeah.
And so the reason why we might not have seen this before is because of some desperate tick needing to feed, and that farmer just got very, very unlucky, but it's never happened before.
Well, some of the articles say that it's probably been around, but we just didn't recognize it.
Oh, okay.
That's the other possibility.
Okay.
Like the Heartland virus.
Yeah.
The next one is from... Did you want to say something?
I was just going to say it's named Bourbon Virus because that's the county where the patient lived, Bourbon County, Kansas.
That's not the treatment. Right.
It's actually in the heartland of America, too, so it gets confusing at some point.
Paul writes, to the TWiV, a great year for TWiV. Thank you for all the good work. An unseasonably warm, drizzly, and foggy 11 degrees in Philadelphia, PA. Thanks for mentioning National Influenza Immunization Week in episode 315 and giving an outbreak update in 316. I read that the flu vaccine for the coming season will be the same as last year, and he gives a link to the Morbidity and Mortality Weekly report on that. In 315, one of you advised that even if the vaccine is unchanged, you should get it as a booster. Revaccination for new strains is one thing, but revaccination for the same strain is confusing to me. No other vaccine has an annual booster schedule. I thought the mechanism of protection from vaccine depended on the establishment of various types of memory cells that hasten and strengthen the adaptive immune response later upon infection. The reuse of prior year strains seems to focus on the strength and persistence of the primary antibody response, making bullets, rather than the establishment of a population of memory cells, making bullet factories. that might be quiescent until activated upon a later infection. Is immunization sometimes more like passive immunization than it is like the body's experience of actually surviving a disease? I got my flu shot this year, but I am not sure why. I reference below an interesting note from Plotkin on the difference between antibody levels as direct and indirect indications of protection, and a couple of sites about antibody persistence. Also, nomination for a pick of the week from the History of Vaccines, a project of the College of Physicians of Philadelphia, a game that goes through the development of society as it fights disease. Illsville.
I went to this game. It's pretty intricate, and it's got 4 eras. I only played for partway through the first 4, and there it is. I diagnosed the first 2 diseases right, and then the third one I got wrong. And you're starting out in the 1600s, and as a game player, you have nothing to do because there's no doctors, there's no vaccines, and so you just have to watch what happens. And I didn't advance on to the next round because I ran out of time. But,
cool. Sounds good.
Yeah, I think your son might like it.
Flu vaccines. The problem is Paul, that especially in the elderly, the memory response is pretty poor. By the end of the flu season, the immunity is waning. That's why it's recommended to get it every year. And in other people, it's not great either, and your immunity probably wanes in maybe 2 years or so. So it's just convenient to have it every year, especially for the elderly, because it will provide them with another bolus of B cells, right? Maybe not good memory, but enough to get you through the season. That's my understanding, anyway.
Yeah,
it's unusual. Yes, you're right. All your points are correct, but this is an unusual vaccine, because it's an important disease to immunize against, but the vaccine could be better.
I just want to point out, I don't know exactly when Paul wrote this, but the article he cited from MMWR was in August, and that was reporting the vaccine conference from the previous spring about the vaccine for this season. So 2014-2015 is evidently the same as 2013-2014.
Right.
And we're just now coming up to the time when they'll be making the decision about next year's flu.
And it's not this year. It turned out the H3N2 component, which is the predominant strain circulating here, is not a good match.
Right. So I'm pretty sure that at the very least the next round of vaccine will be will have that component fixed, as the Southern Hemisphere vaccine right now already does.
Right.
Okay.
So they do different vaccines for the Northern and Southern Hemispheres because just like the seasons are opposite, the flu seasons are opposite. And in fact, the folks picked up on this strain emerging in the surveillance, decided to include that strain in the Southern Hemisphere vaccine. It was too late to include it in the Northern one. So the advice from the CDC has been, for anybody who happens to be traveling to the Southern Hemisphere, if you have a chance, get your flu shot while you're down there. Because that vaccine probably has better efficacy.
Interesting.
But most likely too late.
Don't everyone go traveling to the Southern Hemisphere at once.
Well, I don't know. It's looking at the temperatures for this week.
Helen, you're next.
Okay. Let's see. So we're at Anthony, right?
Yeah.
Anthony writes, Dear TWiV friends, an iron chelator pill called Deferiprone induces the apoptosis death of HIV-infected cells. Another drug called Romidepsin activates hidden HIV and drives it out of its reservoirs. The links below say Deferiprone.
You good luck.
Deferiprone cannot be considered a cure for HIV because these pills eradicate the virus in infected cells. It can't reach all of them. Meanwhile, Romidepsin isn't considered a cure because it can drive the viruses out of hiding but can't eradicate them. Could you explain what you theorize might happen if Deferiprone is ever combined with Romidepsin in patients infected with HIV? Happy holidays. I love listening to your netcast.
I would guess, as you are probably thinking, Tony, that the Romidepsin would activate latent genomes, and then those infected cells would drive to apoptosis by the Deferiprone. That's in theory, right? But you'd have to test it. I don't know if there might be confounding factors that would prevent it from working. The problem is, of course, you have these long-lived reservoirs of latent HIV and stem cells where the virus is not replicating, and so it can't be. Those cells do not undergo apoptosis when treated with the deferriprone, and maybe it can't even reach them because that's actually not. Is this deferriprone specific for HIV-infected cells? Because it's an iron chelator, I think. Yes.
Yeah, that sounds kind of general. It's used apparently to treat thalassemia.
Well, maybe it is specific for virus-infected cells. Yeah, there is a paper here.
Awaiting approval in the U.S.
But that would be my prediction. What do you guys think?
Well, you would also have to have the romidepsin activating 100% of the hidden HIV, and it probably won't do that 100%.
So romidepsin is a histone deacetylase inhibitor. So basically what that means is it allows latent HIV to be transcribed and start replicating. And people are exploring this as a way to get HIV out of the latent reservoir. And one of the approaches is to do that and then hit the patients with antivirals. That would sort of achieve the same thing because then you'd prevent infection. The original cells would die. You'd prevent infection of new ones in theory. So people are exploring that, but not with deferriprone, as far as I know. But I guess in theory that it's sort of a similar thing, except instead of inducing apoptosis, you're just using an antiviral. Stay tuned. We'll see. Dickson, can you read the next one?
Be my pleasure. Carolyn writes, Hello. I have two questions. One a virology question and one a bit more personal. Are virus infections more likely to be species specific than bacterial infections? It seems to me that they would be, since viruses rely so much on the specialized relationship with cells for survival. Okay, we'll hold that. Second question. I'm a bachelor's level clinical laboratory scientist, medical technologist, working in a large hospital laboratory in toxicology and chemistry for the past 15 years, but my love is really infectious diseases, virology in particular, and I'm hoping you can give me some advice. I'm considering pursuing graduate work on a part-time basis in virology. Since I am now, in quotes, middle-aged, I have to consider the marketability of my choice. What are some areas of study that involve virology but are also marketable? I do want to pursue what I love, but I have to be realistic as well. Thanks so much for your advice. I love your show, and even if I never had a degree or work in virology, I will still be listening to TWiV. So we have two questions here. Are virology infections more specific for hosts than bacteria?
The way she puts it is great because she says they rely on special relationships with cells. They're intracellular parasites.
This is very true. This is very true.
Very true. I was thinking about this, and a lot of virus infections that I can come up with are certainly species specific. Not all of them, like polio, measles, right?
Yes.
Adeno. So, and then bacterial infections. Well, they're also human-specific bacteria. I don't know if you can make a general response. I think the point that you make is good, though. There are a lot of specialized relationships. So, I'll say yeah.
The virologist in us would say yes. But maybe we're biased.
We don't have the data. Somebody would have to do I guess what you do is an analysis of pooling all known bacterial diseases and all known viral diseases and trying to somehow search the literature on all of them for host specificity.
Well, the word promiscuous arises here very quickly, in my mind at least, and the first thing I think about is a thing like West Nile virus, which not only infects a wide variety of mammals and vertebrates, but also infects cold-blooded invertebrates. Yes. So there are ranges of infectivity which may not be related very much to the kinds of organisms, but rather to the specific evolutionary tract that each one took during their eons of development. How's that for a non-answer?
Yeah, I mean, that's one virus, though. There are many others that are species-specific, right? So it's a hard question. But
I mean, you worked on one, right? The polio virus, which appears to be...
Yeah, Dickson, I just mentioned that. You didn't listen to me.
I was busy thinking of my other answer.
I think the point, though, is that you can learn something from thinking about this question, right?
Mm-hmm.
And what is that, Dickson?
That you have to think more about this in order to come up with a more complete answer.
I'm going to ask my colleagues on TWiV this.
Yeah, no, you should. You really should.
That's a good idea.
I'm trying to find my TWiV show notes here. In the meantime, why don't you guys answer her
second
question?
Yeah,
second question.
Well, more difficult.
What to do to follow up on her interest in virology and maybe make that marketable?
Well, what if she became a specialist in PCR diagnosis, for instance, for viruses in the laboratory as a medical technologist?
Yeah, I was thinking of that, but I think she sounds like she wants to do research, right?
Still narrowly focused, right? What about opportunities in industry?
Well, it might be that she might be able to look for something in industry. If she's got good hands and is, good at thinking and taking good notes and so forth, that might be really good for an industrial position.
Yeah.
The employment prospects are not good.
Yeah.
I mean, it pains me to say this because for years I encouraged people, if you love science, you should go into it. But I've come around to the view that it's now a bad idea to continue encouraging people to go into especially graduate work. It's a huge, huge commitment. And at the end of it, you enter an enormous pool of people who are competing for a small and probably shrinking number of jobs.
True, but there is one activity which she should be good at after acquiring this knowledge, and that is teaching.
There are a lot of people with advanced degrees in the sciences who are thinking exactly the same thing.
Yeah, I know, but there are lots of turnovers, and this is an aging population.
That argument has been floated for the past 40 years, and it keeps not happening. The problem is that each PI trains a few dozen people, and that PI eventually retires or dies, and now that's one job opening for a few dozen people, and the ability of our economy to absorb these people in a way that uses their degree is just maxed out. And obviously we've talked about this on TWiV, and there's an enormous amount of discussion online about it. That you've got all these people in indefinite postdoctoral positions floating around trying to decide what to do next. And
well, I'm thinking more along the lines of, not that she should go get a PhD, but what if she just got some courses toward a master's or got a master's degree?
Yep.
Then I think that would make her marketable in the biotech or pharmaceuticals.
I agree, because she's already a med tech.
Or she could head a laboratory, a diagnostic laboratory.
And I agree. I was going to say the same thing. Get a master's in focused on virology, and then I think you could parlay that with your past experience into a, kind of a parallel level position.
Or a managerial.
That actually is a really good idea.
Yeah.
Using the degree as a way to get promotions within your current field, because medical technologists, there are jobs for those, as I'm sure Caroline is aware.
You bet.
And there will continue to be jobs for them. And one with a master's degree in virology is looking more like management material.
Yeah.
So that's a definite possibility.
Yep.
All right, Kathy, you're next.
So Ramon writes, recently you talk about the sexual route of infection and HCV. I thought you'd be interested in this article. And then below that, there's this link to hepatitis C virus antibody dynamics following acute HCV infection and reinfection among HIV-infected men who have sex with men. And then it looks like this also came in from another person, Thomas? Or it's a little goofy.
Oh, no, there's an editorial commentary by...
Oh, and that's by...
That's right, Berger.
Yeah, this same guy. Ramon sent both, yeah.
Okay, so... I only just now pulled up these papers.
I couldn't actually access them.
Oh, if you just put in the PubMed ID number into PubMed, they pop right up.
Yeah, but you can't get the whole article. You just get the abstract, right?
Oh, I can get the full final text. Yeah.
It's a southeastern Michigan thing.
Yeah, yeah.
Anyway, what I did is I looked. So that we mentioned the possibility of sexual transmission of Hep C before, and that's what Ramon is keying on here. So I went to Fields, and it says here in the HCV chapter, the frequency by which HCV is transmitted sexually is controversial. and they give a lot of evidence about, in some studies, there's been absolutely no evidence for sexual transmission. On the other hand, HCV infection occurs in persons acknowledging high-risk sexual practices, and there are multiple outbreaks among HIV-infected men who have high-risk sexual exposures with other men. One speculation is that, as with HIV, the risk of sexual HCV transmission is greater during the acute phase when viremia peaks and prior to the formation of antibodies. So there's clearly a cohort of people who do transmit HCV sexually. And so the comment, did you get access to the commentary, Kathy?
Yeah, I have that too.
What are they saying? Basically, we have to screen people who are not just IV drug users or something, or let's see here.
You get the editorial commentary up.
Because I clicked the direct link and I didn't, but maybe that was a mistake.
Current guidelines recommend HCV antibody testing in patients presenting with HIV infection, and HCV RNA testing should be performed in those with a positive antibody response. And then they talk about there's a gap between the recommendations by guidelines and the actual clinical practice. The majority of patients were screened by anti-HCV testing within 3 months after they first presented at HIV primary care clinics. The follow-up HCV screening modalities didn't follow the guidelines in a substantial proportion of the patients. Only 56% received additional HCV tests. There's still need for improvement of HCV screening, especially in the setting of AHC. I don't know what AHC is.
Yeah, I'm trying to find that too. It doesn't define it.
Yeah, unless it's... Oh, there it is. Acute hepatitis C infections.
Jeez.
Yeah, okay. Could they not spell it out? But whatever. These novel data on anti-HCV dynamics in HIV men having sex with men are highly relevant because they support a broader use of sensitive quantitative PCR-based HCV RNA testing.
I think they want it earlier.
Yeah.
Also as well. That's the key here.
Right. So.
Okay. Yes, I was able to go through PubMed. I should have done that originally. I did not. I went to the direct link. Thank you to Ramon. Now, next is Vincent. Vincent, can you read the next one? Sure. No problem. Bill writes, Vincent, this might interest you. Actually, it might interest the entire TWiV team. So,
yes.
It's very interesting. I had not known this, but it's an article in Tech Dirt. Will patents ruin the most important biotech discovery in recent years? And I had no idea. But CRISPR, the wonderful CRISPR discovery in bacteria of a memory-based, nucleic acid-based defense to plasmids and viruses, which can be used in eukaryotic cells to do amazing gene editing, apparently is under a patent dispute. And a variety of companies have been formed. The Broad Institute at Harvard was given a patent on it, and this is being challenged. So they're saying here that, in terms of therapy of patients where patents are important, this is going to be a problem. Although in the lab we can use CRISPRs anyway, so it doesn't really matter to us. But it's quite an interesting problem. And in researching this, I found that the two people who are credited as early discoverers, Emmanuel Charpentier and Jennifer Doudna, got this prize in November for tons of money. What is it called?
It's this brand new prize that.
Yeah.
Oh man, it's incredible!
It's a huge prize.
$3 million.
Yeah.
Yeah.
So they have, and they're all dressed up and fancy with with actors and rich people on stage. Really impressive. Anyway, so this is a big. Do you know about this, Alan Dove?
Yes. I covered a conference at the New York Academy of Sciences where Doudna and Charpentier got another award. And that was before they got the $3 million. It was the Jansen Award, which is pretty prestigious. But it doesn't come with that big a check. And they talked about their work, and there were a bunch of other speakers there talking about applications of the technology. So Doudna, Charpentier, and then this fellow at the Broad, Zhang, are the three people who have filed patents on CRISPR. Zhang and Doudna apparently co-founded a company called Editas, and then when Zhang's patent issued, Doudna left the company and took her patent to another company. Her patent is pending, and then Charpentier, meanwhile, sold her patent to another company. So you've got potentially 3 companies with 3 different either issued or pending patents. Yeah.
I asked my colleague Saul Silverstein, who is well versed in these patent issues, having had one, and he said the lawyers will win out.
Yeah.
I like the first 2 comments under this article. The first one says, "Good thing this syndrome didn't begin in an earlier era. We'd all be dead." And the next one says, I can't remember who said it. It might have been Bill Gates, though I'm pretty sure it was one of the major players in the early computing world. Basically, that if today's IP laws had existed when computers were first entering the scene, the computer as we know it would not exist.
There you go.
All right, Alan, you are next. We're up to Nick, I think, yeah?
Yes, yeah. Nick writes, "Dear Twivologist, short-time listener, first-time writer. As someone raised in New England, I'm naturally a huge hockey fan. Go Bruins. As someone who's an aspiring biologist, I'm naturally pro-vaccine. If you've been keeping a close eye on the news, you know where this is going, and I mean very close, because not many people seem to be talking about this problem or the NHL in general. There is currently a mumps outbreak in the National Hockey League, with teams all across the country having infected players and coaches. What is further alarming is that reportedly all of these players have not only had the MMR vaccine, but some of them have recently received a booster as well. Can you help explain why the vaccines are failing? Is it just regular old vaccine failure, or is it something more worrisome? I also want to thank you guys. Your team has been a regular inspiration to keep on pushing on as I attempt the arduous process that is applying to graduate school. And yes, at your behest, I did apply to ICON as well as Vincent's current employer. Weather report, ditto to Vincent." This letter, obviously, we're a little behind on our letters. This mumps outbreak happened a little while ago, a few months ago.
The vaccine isn't failing. There is. I looked up in CDC. There's an 88% effectivity of the vaccine. One dose is 78%. So, and when you have a team that is all together and traveling, and 88% is, you're going to have an outbreak.
Yeah,
I think that's basically it.
Right.
This was one of your picks, Dickson, tongue in cheek, right?
See that?
Yeah, I thought when we talked about this before that Rich had commented that there was some evidence that the mumps vaccine is a little bit less effective than had been hoped, or that the coverage wanes.
In my podcast in Georgia, one of the people I spoke to was talking about that, and that he's working on making an alternative mumps vaccine.
Oh, right.
There's some waning of efficacy, yeah.
Yeah.
So that may be part of it. All right, now, Dickson, this next one is quite long. Are you okay with that, or do you want me to give it to someone else like Kathy Spindler?
Shall we enlarge the text?
Stop it.
It won't transmit to him because he doesn't...
Effie writes.
Oh, okay.
"My name is Effie, and I'm a master's student at the Weizmann Institute of Science in Israel, and a devoted listener to your wonderful podcast. Also, I am currently taking your course on Coursera and enjoy it tremendously, I must add. Actually, I like the course so much that I decided to dive into the world of virology, though my own work is in neurobiology lab. Recently, my current work incorporated a new tool to decipher an enigmatic question with regards to a neural circuitry, which involves certain sexual behaviors. Funny enough, this tool is none other than the H129 Delta TK herpes virus, which was developed by the talented people of the David Anderson team at Caltech. Using the famous Crelox system, we can trace the circuit in the forward direction, anterograde, within the circuit. I have a small question which I could not find an answer to in the literature. I would be grateful if you could suggest a possible explanation. As a security measure, the authors of the paper and developers of the virus knocked out the tyrosine kinase sequence from the original vector, stating that without it, the vector will not be able to replicate at the ganglion. However, I can tell you that the receiving animals show serious illness, and the virus still holds the capacity to infect and jump from synapse to synapse. It must, otherwise it won't be using it. Therefore, what exactly is the purpose of shutting down the TK component? Does it mean the virus can infect but not replicate? I am a little confused on the issue. What happens to the animal once it's infected? Does it carry the infectious particle into the saliva too? I am asking that because the animals often become rather aggressive a few days after the injection or jumpy, and I'm wondering if there's a chance that I'll get infected from a bite. Is it possible? The correct use of this tool is quite important for my current work and my life, and hence I bothered you with the email. Nevertheless, I truly hope you weren't discussing this on the subsequent lectures in your course on Coursera. I still haven't completed it yet. If yes, my sincerest apologies for wasting your time with the email, as I'm sure I'll get to this lecture soon.
You can skip the next part.
Okay, fine.
And just go to again.
Again, thank you, Vincent. Again, I want to thank you for all these wonderful and enriching hours of podcasting. Sincerely, Effie.
So I asked Lynn Enquist, who's the expert on neural tracing using herpes viruses. Would you like to read his response, Dickson?
Sure. Lynn writes, is that the one you want me to read?
Yes, sir.
I helped Anderson build this HSV-1H129 recombinant. We send it out to neuroscientists as part of our NIH National Center for Neuroanatomy with Neurotropic Viruses grant. I have considerable experience with this virus. The thymidine kinase deletion, not tyrosine kinase, is the main selective feature for the Anderson virus. It is not a security measure, ! An HSV strain defective in TK will not replicate in non-mitotic cells like neurons. It will replicate in transformed tissue culture cells since they are always dividing. This is how we grow the virus for distribution. When this recombinant infects a Cre-expressing cell, a silent TK gene built into the virus is expressed and the virus replicates. Since Cre recombination is not reversible, the virus that is produced after Cre recombination continues to replicate and spread among synaptically connected neurons. Ideally, as I said before, in the absence of Cre, this recombinant virus will infect but will not replicate and make particles. It should be essentially avirulent, completely attenuated. If the animals being injected are getting sick, then there is at least two possibilities. 1. The virus is being injected into a site where there are mitotic cells and the virus can replicate even if it is TK-. 2. The virus is not TK-. A TK- virus is resistant to acyclovir, which blocks replication when TK phosphorylates the prodrug. TK- viruses are sensitive to acyclovir while TK+ viruses are resistant. If you are injecting HSV into a mouse brain, there will not be any virus in the saliva. Lynn.
I think I got the right guy, huh?
Hey.
Yeah.
Absolutely.
So, Effie, that's what you need to do. Check your virus with ganciclovir. Or acyclovir, whatever.
Yeah.
And it's not tyrosine kinase. But I think it's funny. He said this virus was made by so-and-so, and Lynn said, no, I made it with him. He probably made it for him. Okay.
Lynn also added that if you're injecting HSV into the mouse brain, there won't be any virus in the saliva.
For sure. Thank you, Dickson.
You're welcome.
I know that was difficult. Kathy, you are next.
Piece of cake.
Okay. Dennis writes, good news on vaccination rates and public opinion supporting vaccinations in Australia, up from 53% in the 90s to over 90% now.
Outstanding.
There's a corresponding drop-off of funds to anti-vaccination snake oil salespeople. And he sends the link from The Guardian that just says that Australia's anti-vaccine movement has a decline in membership. And Dennis writes, it will reach 22 Celsius here today and very sunny in Santa Cruz, California. Best to you and the team, Dennis.
Cool.
I think it's good, but I wouldn't get too excited. I mean, these anti-vax people are really very persistent and stubborn, and it's going to be a while before we can really make an impact.
But if that's really true, that, childhood immunization rates had gone to as low as 53%, and that now it's better.
That's great.
Or opinion had gone as low as 53%.
Yes, it's opinion, right? Yeah. So I think it's great. No, don't get me wrong, but I'm just saying, don't get complacent. We have to keep fighting this.
Remain vigilant.
Yes. It's my turn. Is that right?
It is.
Judy writes, thought even the listeners might find this interesting, and she sends a link to an article. It's very cool. As usual, thanks for all you do and keep my brain working. And Judy is a retired high school science teacher in San Diego, where it's 18 degrees C, 64 F, 10% chance of rain, cloudy winds, 3 miles per hour from the northeast. And this is in Now I Know, a site called Now I Know, and it's called the smallpox boat. And it's an article about. So everyone knows that Jenner developed or showed that immunization with cowpox could prevent smallpox in the late 1700s, and this was a big deal because smallpox was a serious disease, but they couldn't figure out how to get the cowpox to Europe because we didn't have airplanes, and the boats took a long time, and how do you preserve the virus? So this guy Balmus came up with the idea of sending over infected people, and they would infect each other during the trip, and then it got to Europe, and there, bingo, you got the virus. So
do you mean Europe or from Europe?
From Europe.
Yeah, but Jenner was in England, right? For some reason.
Oh, I see what you're saying.
Sorry,
Got it.
I forgot.
Okay.
I thought he was. I'm just thinking
Yes.
he was in the U.S. where all the great science was done. Sorry, I'm just kidding. Don't get pissed off. Anyway, so I couldn't believe this. So I wrote to McFadden and Condit. I said, "Is this true?" And Grant replied. He said, "Yeah, it's essentially true. The live vaccine chain transmission on boats using serial passage in young volunteers was the key to getting live cowpox-derived vaccine to the New World. Once the boat landed, the skin of inoculated cows then took over the job of propagating the orphan-mediated stock of new vaccine that had arrived in the arms of the orphan donors. These passages were very poorly documented. Now you can see why it has been so hard to trace the original lineage of vaccinia virus. There are still debates about what exactly the progenitor virus was, i.e. horsepox, that then evolved through the skin of inoculated people and cows to become what we now call vaccinia virus." And Rich Condit agrees that this is all good and correct.
Right. And it was a way to get the virus from Europe to the Americas.
Yeah.
Right.
Yeah, I had it backwards. Shall we keep going? One more round?
You bet.
Sure.
Good. All right. Alan, you're next.
UA writes, I was just listening to the very latest episode of TWiV. Dickson and you raised up now, when he says very latest, we're way behind on our letters. So that could have been a while. Dixon and you raised a non-virology question as to why there are currently only 4 serotypes for dengue virus, or more broadly, why many viruses have a given number of serotypes. However, as far as I know, the 5th serotype of dengue was reported in 2013, and provides a link. With regards to why we haven't seen the 6th serotype of dengue, it is probably a matter of time in the context of evolution. I'd like to quote a sentence from Field's Virology. Based on comparative sequencing studies using the hemagglutinin molecules, it has been postulated that the influenza A viruses diverged about 2,000 years ago, and the influenza B and C viruses about 4,000 and 8,000 years ago, respectively. If we went back to 2,000 years ago when there wasn't influenza A, we might have asked a similar question of why there are only influenza B and C viruses existing.
Well, but look, there are tons of influenza A viruses, and probably even
Right.
back then. So that's the key here, that even given the time, there's still way more flu than measles or polio or dengue. So,
Sure.
And I had a guy here, an applicant for our PhD program, and we talked about this, and he agreed. Yeah, we just don't know why flu HA, for example, is so variable, and others are not.
HIV, yes, that's right.
Back on this idea of the 5th serotype of dengue, that's not completely widely accepted, I think. When you look at the article, what is maybe the 5th serotype has some similarity to the 4th serotype. And among the dengue strains, there's a lot of divergence when you get to the sylvatic strains from the urban strains. And so I don't know what the definition, what the taxonomists, the ICTV, would say has to be the number of, changes or percent non-similarity or whatever for it to be a new serotype. But I don't think it's completely accepted.
We have some dengue aficionados listening. Why don't you weigh in? Let us know what you think about Dengue 5. Dickson, there's a long one here.
I think it would be a good name for a science fiction movie.
Dengue 5?
Yeah.
Sure. Dickson, can you do this long next one?
I just lost the whole thing right here in front of me.
I'm sorry.
Me too.
Oh, well. Who's next on the list? Kathy.
Sure.
Yeah, go ahead.
Anthony writes, "Thank you for covering 2 letters from me on TWiV 319. The quote from Lin Fo Wang is from the email that I'm forwarding now. I don't know what website was being looked at that resulted in the high maintenance verdict for opossums. That's certainly so compared to cavies, but the American marsupials require no more care than a domestic cat, and certainly much less than any non-human primate. So I'll just do an aside. Last night I Googled again, and I got the same site, which is a link that says possums as pets. High maintenance does begin to describe captive possums. They are not meant to be kept as pets. And I swear to you, it is the most expensive, da-da-da-da. So that's where I got it.
Right.
Okay. Back up to in TWiV 296, the Lin Fo Wang episode, there's talk of establishing a colony of fruit bats and the difficulty involved. That would be multiplied many times over for a researcher in the United States. Possums seem to have a similar resistance to viruses. Someone here could lay the foundation by finding out what, or if, that is, once a cause has been established, it could be checked to see if the same mechanism is in operation with bats in Asia and Africa. I mentioned roadkills not as an optimum source of data, but as one very easily available and at basically no cost. In addition to the Virginia possum, common throughout the eastern U.S., there's a much smaller, around rat-size, South American species that's already domestically raised for the pet trade. Possums can be maintained quite easily in tall pens. All bats clearly should have lots of space to fly. Lin Fo Wang mentioned that his colony would not have room to fly. I believe that you replied how this would make it difficult to discover if the high rate of metabolism during flight was what provided the resistance to viruses. Possums don't have any high-power aerobic capacity, but they do seem to have the same resistance. What they do have in common with flying mammals is the opposite: low body temperature, always for possums, and when not flying, for bats. Most small bats are, for all practical purposes, impossible to maintain in captivity because they consume huge numbers of flying insects. As they eat fruit, the flying foxes are easy to feed, but these large animals truly are high maintenance. If any researcher was establishing a colony of possums, I'd guess that few would take any notice. How would the public feel now about numbers of bats being brought from Africa to the U.S.? I think that the New Jersey State rabies spreadsheet that I sent you has question marks. The data comes from animal control officers responding to complaints. A question that I have is whether possums afflicted with rabies simply crawl into a hole and die without causing a ruckus and so are not counted. Even the supposed symbiosis of fruit bats with viruses, parentheses, symbiosis is a misleading term, for most tend to think mutualism, not commensalism or parasitism. Anyway, back to the main sentence. Even the supposed symbiosis of fruit bats with viruses seems to need a lot of work to establish. Fruit bats require a lot of food, and their only defense is flight. Like birds, they won't look sick for very long, if at all. When to any degree they lose their edge, they die or are killed. It seems that there haven't been any longitudinal studies to find out how long any individual bat has a virus. Could an infected bat be like a person with HIV? Bats in the U.S. were thought to be carriers of rabies, not killed by the virus themselves. That seems to be wrong. I was Googling Charles E. Ruprecht, for he is one of the lead authors here. He gives a reference. Bats, Emerging Infectious Diseases, and the Rabies Paradigm Revisited. It is stated that, more recently, the asymptomatic excretion of rabies virus in the saliva of experimentally infected vampire bats, which survived the challenge during at least 2 years of observation, was documented again. So he's implying that they're persistent. But the cited research, despite what one might expect from the title Salivary Excretion of Rabies Virus by Healthy Vampire Bats, states that, quote, rabies virus was not detected in the saliva of any of the 11 animals that succumbed, somewhat early, to rabies challenge, nor in the control bats. In contrast, virus was detected early, and only once, days 6 something and 21, in each of the 3 animals that survived rabies challenge and remained healthy for at least 2 years after challenge. At that time, even vigorous dexamethasone and cyclosporine administration failed to provoke further viral excretion. This appears to show that bats once recovered are no longer carriers, and not that apparently healthy ones are. Many thanks to you and your colleagues for TWiV and the other great series of podcasts. Anthony.
So Anthony, have you actually raised possums?
Wow.
He's an advocate.
Yeah.
'Cause I'm kind of the assumption I would have is that they are high maintenance.
Yeah.
They always struck me as pretty wild animals.
Yeah.
But there's a really, I think that's a different question. Would they be high maintenance as pets versus would they be high maintenance as laboratory animals?
Yeah. Well, I mean, clearly Anthony is questioning the value of bats and zoonotic diseases, but a lot of people are studying bats because there's good evidence, despite what he has provided here, that they carry viruses, that SARS, corona originated in bats, that Nipah, Hendra. Yeah. So that's why people are studying bats, and there's just not that evidence for possums, So what's this thing here? Cheeseburger.
That was another
about a lost and found poster for a possum.
That's funny. Not awesome. Very friendly.
So it's somebody put up a cat found poster.
That's funny.
That's 2 pictures of a possum and not housebroken. Found on 75th
really
issues year is called down.
That's pretty funny. Dickson, have you recovered? Do you want to read the next one?
I have.
It's long.
Thanks to you.
It's long, Dickson.
I'm going to give it a twirl here.
Natalie.
So Natalie writes, "Hello, TWiV team. I'm a former chemist in my late 20s that is waffling with the decision of leaving my cushy 6-figure job to go back to school for a PhD because I believe research is extremely important for our society and want to contribute. However, I recently came across an article with some very sobering and compelling reasons for not to pursue grad school and a career in academia entitled, 'A Career in Science Will Cost You Your Firstborn.' Then there's a link to that article. Although the article has a bit of a cynical, one-sided take on the matter, at least grad students aren't going further into debt, and the title is a bit dramatic, I think the points raised and the quantitative analysis are fairly accurate. And to be fair, I know plenty of equally cynical grad students who would agree with the author. To summarize, the article shows that wages lost by opting out of a non-academic job to instead pursue a career in academia could roughly equate to the amount it costs to raise a child, about $200,000. This financial analysis really struck a chord with me because I myself am weighing buying a house versus getting a PhD in the next 5 years. A bit of background on myself. I earned my chemistry degree 5 years ago from a top research university and intended to continue my education with a PhD. After graduating, a bad viral infection, mono caused by EBV, sidetracked me into a career in which working from home while recovering was possible, software engineering. My 2-year battle with mono is also, coincidentally, how I found your wonderful podcast. I've recently been accepted into a PhD program, and though I remain wildly passionate about science, falling into the lucrative software industry has made the decision to return to academia all the more difficult because of the significant pay cut it would require. Money is not everything, of course, but grad students' stipends seems drastically low given the average rent in areas like California and New York. I feel incredibly guilty if I sell out and cite personal finances as a reason for not pursuing science, but the pragmatic side of my brain is struggling to justify a pay cut of at least $70,000 per year for the next 5 years. My friends think I'm crazy for even considering it. It's a shame this is such a hard decision. I think I would make a great scientist, and I fear that other capable graduates shy away from academia for financial reasons too. So in your opinions, is academia still worth it? Is it too late to start an academic career in your late 20s? Please share any advice you might have for anyone wrestling with this, with such a decision. I'm happy to see that grad students and postdoc issues are being brought up for discussion more frequently and hope that such discussions ultimately lead to an improved quality of life for grad students and postdocs and make pursuing a career in academia a more feasible option for those with a passion and aptitude for it. Thank you for all the podcasts, Natalie. P.S. My ears perk up every time Epstein-Barr virus is mentioned in your episodes. Any chance of an Epstein-Barr virus episode? Apologies, forgot the weather. San Francisco, it's 50 degrees Fahrenheit and oddly not foggy for once.
So this article is written by John, who has written to TWiV before.
Yep.
Yes. Well, let me just say that if you can't think of doing anything else in your life, is science is the only thing you want to do, then you should do it. But only if you can't think of doing anything else.
If you can think of doing anything else, particularly at this point, I recommend staying with your software engineering job.
Or if you're doing something else.
The one thing I would add, though, is for this particular person, for Natalie, if you're in software engineering and you have, obviously you probably have pretty good coding skills at this point, you might look into bioinformatics PhDs.
Absolutely.
Mm-hmm.
That is one of
God.
very, very few, maybe even the only graduate PhDs that has real employment prospects right now, and probably still will 5 to 10 years from
great
now.
advice.
Sure.
Perfect.
It's the 2015 version of plastics.
That's right.
Yes.
Yeah, but
bioinformatics is my advice. If you have the capacity to do that, and it sounds like you probably do, then that would be something to look into.
Sure, but I subscribe to Chemical and Engineering News, as maybe some of you do also, and I see their job opportunity sections every 6 months or so, and chemical engineering seems to always come out up on top someplace, not just for undergraduates, but also for graduate
He
students.
doesn't want
as
to be
well.
a chemical engineer.
I understand that, but
He wants to be a virologist, but I think that bioinformatics approach, because you could get some experience so you'll understand the field, but then really do. This is all we talk about in the graduate program. We're not teaching our students bioinformatics, and people doing that are doing well, but they don't know the science also. So if you can do both,
how about that?
Yeah, so here again, I would say more and more places are starting to have serious master's degrees where you could potentially get some bench science work in a virus lab and bioinformatics coursework that could be.
Now,
just the
bear in mind, any kind of demand for employees with particular skills will eventually be satisfied.
Right.
And when that happens, if there's still a stampede of people going into bioinformatics thinking that they're going to get great jobs, then you'll just mimic exactly what's already happening with PhDs and the rest of biomedical science, and you'll have this huge group of people who are unemployable.
Yeah,
I think we have time though, because.
But I think we still have time. These are still people who are highly desirable, and the reason they're so highly desirable is because academia and industry both need them in large numbers.
Sure.
Right.
And
There's all these huge data sets being generated.
And everyone's genome is going to be sequenced, and we
Yeah.
have to have the software to deal with that, which we don't have yet.
Maybe the next big field should be called data fracking.
Yes.
Good title, Dickson. All right, our last one for today. Jacob writes, Dear TWiVers, thanks for the great episode covering the recent dengue vaccine trial. The antibody-dependent enhancement theory that has been put forward to explain the increased risk of developing severe dengue during secondary infection is immunologically interesting. If this was a general mechanism, it would be highly unlikely that it only affected infection with dengue virus. Many viruses are capable of replicating in cells bearing FC receptors, and the spectrum of antibody specificities in a given individual at the time of a specific infection likely offers lots of antibodies that can bind an infecting virus with suboptimal affinity and thus potentially give rise to antibody-dependent enhancement. It has, however, not been obvious from the study of infectious diseases that this is occurring. On the contrary, most suboptimal antiviral immunity, either from vaccination or infection with similar viruses, tend to result in a milder disease course, i.e. some immunity is better than nothing. Even for dengue, the data supporting a causative mechanism for antibody-dependent enhancement in severe disease is not clear in my opinion. If such a mechanism truly exists in vivo, I would think that it should have been visible in this large vaccine trial. There are also a number of other publications that have failed to find a clear connection, and he gives a link to a PLOS Medicine paper as an example. As an aside, it is always striking to see how low the mortality rate really is in well-conducted dengue trials. Jacob is at the Karolinska in Stockholm. Yeah, there's a lot of uncertainty about the antibody-dependent enhancement. It has, to a certain extent, been replicated in mice, though. We talked about a paper a long time ago in which dengue in mice, and this was replicated using antibodies. But, of course, they're not people, I understand. I sort of agree with these reservations. You would think it would be a more general mechanism of viral immunopathogenesis, but it's not. Good point. All right, that will bring us to... let's see. I don't know. That's about half of the emails, right? Maybe 3/4 being optimistic.
Half of the ones that you pasted in, are there more?
Oh, there are just 5 more, I think. But, it's not too bad.
Okay. That's good.
But, they come in.
It's like doing the wash. It always seems to fill back up again.
It's okay.
No matter how empty you get it.
Is that right, Dickson?
I think so.
Let's do some picks. What do you think?
Absolutely.
Dickson, do you have a pick?
I do.
Oh, yeah.
I have a pick.
Nice.
This is a macro photography pic. And the moment I stumbled onto it, and it was simply by accident, I went to the first picture and I saw this big, gigantic eye looking back at me. I had to see more. And so you keep looking at these, and the more you look, the more you see. It almost looks like you're looking at fractals. You know those colored fractals that you can get in the books called
What is a fractal, Dickson?
It's a geometric figure generated by a simple formula. And it's a mathematical thing, but
This cat doesn't look like a fractal.
No, it certainly doesn't. But they're you see things up close; they don't look like the things they are from a long distance. So it gives you a new perspective, and that's what we're looking for: new perspectives.
It's cool.
These are
nice.
Gorgeous photographs.
These are really, really lovely.
Yeah,
I like the bug eyes are cool.
Yeah.
Look at this guy with the last one, the red vein darter. Gosh, he's got all these. How many eyes does he have, or she? Oh, it's covered in dew. I see. Those aren't eyes.
Right.
Oh, right. Yeah.
Nice.
Right.
Well, for once, you have a pick, Dixon.
I know I had a pick
I'm
last
just
time.
kidding. I know.
You know, you're picking on me.
I am.
As the pick.
Kathy, what do you have?
Well, I picked this Octopus Supermom story that was in Science last July, and it's a video where at the Monterey Bay aquarium, they monitored this octopus over 4.5 years.
Holy cow.
And identified her as the longest egg brooding individual ever recorded. The 14 months was the previous record.
I'll be darned.
And so it's a Granellodoni boreopacifica is the way he said it.
Of course it is.
And they were able to show that it was her because there are these scars on her arms, and so they knew it was the same one, and the eggs kept getting larger, so it wasn't that she'd laid another set and they'd missed it or something like that. It's an incredible video, about 3 minutes long, and well worth watching.
1,300 meters is below the ocean surface.
That's pretty deep.
Yes.
Wow.
Yeah.
So it should also be noted that all female octopuses, once they lay their eggs, they die.
Oh, I was not going to tell that part of the story.
I didn't know. I'm so sad. You know what? You can edit it out. You can edit it out.
I'm not.
This is not a live show.
Well, no, but it was a
So
spoiler.
sorry. Kathy, how was I to know this?
No, I know.
So this is an Octomom worth talking about.
Right.
Yes, exactly.
Yeah.
That's right.
Yeah, it's pretty cool. Very impressive. I wonder how often that happens.
What?
But I don't think much is known about this. These deep ocean species have been very hard to study, obviously.
Yeah, sure.
So, and they just happened on this one at their research site, and then the next year it was there again, and they just kept monitoring it.
Did you ever see the vampire octopus by any chance?
Mm-mm.
Nope.
It's a blood red. It's quite remarkable, and it's found very, very deep as well. And,
Yes. Yeah.
What do you have, Alan?
So I have a book that it's a short book, very short book, kind of a I don't know. I think it's called a Kindle single or something. Can breeze through this thing in a couple of hours. It's called Boom, and it's by Tony Horwitz, who's written a number of other books. But this is about the Keystone XL pipeline, which you have probably heard of. Very much in the news, very much in the political news. So what Horwitz did was he got on an airliner and flew up to the subarctic area in Alberta where the oil sands are, toured around there talking to people, and then traveled down the proposed route of this pipeline. And along the way, he talks about what the current pipelines do, and what's driving all of this, of course, is our insatiable demand for oil. And he interviews people all along the way who have very divergent opinions about what's going on. And I thought it was just a really good coverage of this major issue that is now in the process of being decided.
Right.
Wow, gasoline is ridiculously cheap.
You think they're gonna? Do you think they're gonna override Obama's veto of this when they actually reverse the decision that was made in the last Congress?
Maybe. I mean, there's there are already pipelines along this route that are doing a lot of this same stuff, and we need the oil.
They just had a big oil spill up in North Dakota in the Yellowstone River.
Yeah.
Right.
So, when that hits the news again and again, haven't we learned anything from any of this?
And the alternative is we transport it on railroad cars, which then get into accidents and have oil spills.
It's messy everywhere you look.
The fundamental problem, and Horwitz, I think, actually does a pretty good job explaining this. The problem is on the demand side.
Yeah.
I mean, yes, there are problems with this pipeline, and there are some very rational objections to it. And I came away from the book undecided on this issue.
Really?
When I had actually kind of gone into it with a particular opinion, and I came out of it thinking, well, this is... I'm not sure that I'm strongly of one camp or the other on this.
Dickson, you're not involved because you have an electric car, right?
I have a semi-electric car. No, it uses gas. But my next model will burn... won't burn anything. It'll create water from hydrogen, and I'll be able to drive anywhere I want.
And how is the hydrogen made and bottled for you?
It's split off from water by solar panels in a special device, catalytic converter.
Okay. So who makes the solar panels?
You keep going, Alan.
He will.
Yeah, exactly. I will keep going.
It's a one-time only expense of carbon.
Energy, it's coming from somewhere.
No. Of course, it's not a perpetual machine, but I think solar panels are a good substitute for what we're doing now.
They could be, yes.
My link, is something of mine, and it's spring. I just started my virology course. And it's a little different every year, so you can... I record all the lectures on video. They're all on YouTube, so you can go watch them. Virology Lectures 2015.
This is great, Vincent, because I like the fact that now your syllabus is here. I never saw your syllabus before. You've got the reading links, and the YouTubes will be so much quicker and widely accessible, at least for me, than going through iTunes U. I mean,
Yes.
I download them, and then I'm like, where are those on my computer? And it's different on my iPad. And
I decided to put it all on YouTube this year, and it's all here. So, and I've been revising quite a bit. And the other cool thing is that, in September, our new edition of the textbook is going to be published, but many of the figures are done, and I've been using those in the lectures, so they're all updated and
lucky them.
Very pretty and fixed and so forth. So check that out. We have 2 picks of the week. One is from Ramon, who contributed the article on HCV transmission earlier. This is a Scientific American. It's just a little image of the week. It's called Vaccinate, Do It for the Testicles. I mean, this is just because, mumps infects. This is an image by Katie McKissick, and she draws pictures of measles and mumps and has little cutesy comments. And one of the symptoms of mumps is testicular swelling, and she writes, I don't have testicles, but that sounds truly horrible.
And then she comments on all of them that they're very dangerous for pregnant women, and so that's
definitely right.
Yeah, it's a good poster.
It's cute. Yes. And the other is from Sandra, and I will let Kathy Spindler read this one.
Okay, so this is from Sandra, our friend in Texas. I would recommend you listen to this 15-minute story without reading anything about it first. The ending will be that much more shocking. I listened to it a few nights ago and was so impressed with it that I emailed Kathy about it, only to find out that she too had heard it and thought it was great. And this is from Sandra. It's as of 10:30 a.m. Central Time, Thursday, January 15th. It is 43 degrees Fahrenheit with 5-mile-per-hour winds out of the north-northwest, humidity 54%. First sunny day we've had in a while, which is unusual for Dallas. And so this is a link to The Moth, which is something you can find on NPR. There's also podcasts. And the link that we found takes you to the Moth page where there are a number of episodes. So you have to know to look at the one that was released on January 13th, and it's called a special live edition, and it was recorded at a Moth story slam or whatever with all scientists speaking. And the last one in the episode, which starts at 38 minutes 50 seconds into the recording. This is another thing I had a hard time doing on iTunes, but you start playing it and then you can scrub ahead to 38 minutes, 50 seconds, is Mary Claire King. And I'll just tell you that it's a phenomenal story. And I listened to it, I think, at least 3 times so far, and I would listen to it again. I haven't listened to it in a couple weeks. Just amazing. So for those of you who might not know, Mary Claire King is a geneticist who discovered the BRCA1 gene locus that causes hereditary breast cancer. And she's also used DNA genetic strategies that reunite missing persons or their remains with their families in South America. And in 2014, she won a Lasker Award for Special Achievement for both of those things. And this story is just, if you don't do anything else, when you finish listening to this podcast, go find this link and listen to this story.
All right. Thank you, Kathy. Thank you, Sandra. And that will do it for TWiV 322. You can find it on iTunes and at twiv.tv. And send us your questions and comments. We love them. We really do. TWiV at twiv.tv. Dickson Despommier can be found at verticalfarm.com. Thank you, Dickson.
What a pleasure.
The world of vertical farming is heating up, right?
Indeed it is.
Good. I can tell your clothing is improving. You must be making a lot of money.
No, not even a little bit. Come on, that's not the idea here. No, but many new vertical farms have opened up in the last 3 or 4 months.
Great.
Very encouraging. And in fact, one company has now got 5 branches, so to speak, to their original facility in southwestern Michigan. They now have facilities in Ohio, in New York, soon to be in New York, and in Detroit. So quite a good sign, actually.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. I enjoy the all email shows.
Yeah.
Alan Dove is at turbotplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.